My name is Emmanuel Fajemisin. I'm a PhD candidate of the Medical Biotechnology and Immunotherapy Research Unit at the University of Cape Town, South Africa. So special thanks to the Beyond Science Initiative for the opportunity to present in this conference. I'll be giving a presentation on SNAP-tag antibody conjugates targeting CD64 overexpressing cells in chronic diseases. An introduction: macrophages are monocyte-derived cells from bone marrow, which mediate cellular physiology and are also involved in the pathogenesis of several chronic diseases as listed here, which could include infectious diseases, inflammatory diseases, neoplastic diseases, and age-related and metabolic diseases. Currently, globally, chronic diseases account for 3 of 5 deaths, and these dysfunctional monocyte or macrophages could exist in the following forms. We have dysfunctional pro-inflammatory macrophages, which are mostly involved with chronic inflammatory diseases, or otherwise known as the M1 type macrophages. Then also we have the tumor-associated macrophages, or M2, which are closely linked to myeloid-derived suppressor cells, which are actually dysfunctional monocytes, which plays major roles in cancer progression and metastasis. And also, we also have pathogen hosting macrophages, which are actually macrophages that are hosting parasites, which include viruses and pathogens, and they are responsible for infectious diseases. So the current clinical gap, which is the aim of my research, is to develop a clinical agent that can selectively target and eliminate these dysfunctional monocyte or macrophage cells without affecting the normal cells. So as we all know, normal cells actually have normal receptors, which in certain cases, especially in diseased conditions, these receptors are overexpressed. And so the most standard clinical intervention involve chemotherapy, which are conventional drugs and toxic chemical agents, which actually induce DNA damage in these cells. Example is the pentavalent antinomas, which is used in infectious diseases like leishmaniasis, and in certain breast cancers, we have the docetaxel cisplatin doxorubicin, which are actually chemotherapeutic drugs for treatment. However, because they are non-specific, so they have the limitation of high toxicity, drug resistance, and bioavailability issues, and they could also be expensive. So therefore, to overcome this limitation, targeted therapeutics are new solutions, and in this case, our lab-used therapeutic agents, which are recombinant proteins, which contains antibodies attached to toxic agents or toxins, basically, which binds to overexpressed receptors on diseased cells, and by internalization of the toxic domain, they actually cause selective cytotoxicity. And we have been able to work with recombinant immunotoxins, human cytolytic fusion proteins, and antibody drug conjugates. And the advantages is that there is controlled release of the warhead, there is reduced cytotoxicity and systemic side effect. Specifically for the case of this research, I'm looking at exploring the CD64 receptor, which is an overexpressed receptor on monocytes and macrophage. So this CD64, otherwise known as the Fc gamma receptor, is a 72-kilodalton transmembrane glycoprotein, which has a strong affinity and internalization for gamma immunoglobulins or its immune complex. And it's been upregulated with pro-inflammatory cytokines like interferon gamma, especially on the CD64 receptor. Research has shown that stimulation with interferon gamma increases receptor to like 60,000 receptors. And also for macrophages, from a resting macrophages to an inflammatory macrophages M1, there is an increase in the CD64 receptors by 4 to 5 folds. Why is this very important in our research? We have also observed that different antibody that have been engineered have can actually influence the clinical efficacy, as we can see below, that a full-length as we can see, a full-length internalized IgG has a clearance time of 110 hours compared to the smaller domain, like the single chain variable fragment. Fragment variable actually have a half-life of 2 hours, and only the variable domain has 0.05 hours. So in our lab, currently, we are using the SNAP tag to actually deliver diagnostic and cytotoxic protein in vivo and in vitro. So what is the SNAP tag? The SNAP tag is a genetically modified human O6 acute guanine acyltransferase protein, which has the ability to combine with to react chemically with a benziguanine modified substrate in a 1-to-1 ratio. So using that, we are able to engineer the single chain fragment variable of an antibody with the SNAP fusion protein and use that to deliver diagnostic or cytotoxic proteins into cells. And in many inflammatory or tumor antigens that has been delivered include EGFR, EpCAM, CSPG4, and CD64, which is my research. So this is just an overview of the process. We have the SCFV being engineered with the SNAP tag and a reaction with the benziguanine modified substrate. For example, the BGR 700 is actually a photosensitizer, which when conjugated with the H22 SNAP, results in a photoimmunoconjugate. In this case, we have the H22 SCFV SNAP R700, and this can also be used both as a theranostic agent, that's a therapeutic and a diagnostic agent, when internalized in a cell under visible light. Then also a drug like the auristatin F, which is an antimitotic drug, can be used to generate an antibody drug conjugate, H22 SCFV SNAP or IF, which can actually induce cell apoptosis when internalized in a monocyte or macrophage dysfunctional cell. So this is an overview of the experimental methodology. The in silico design of the anti-CD64 SNAP plasmid was done using genetic engineering and also basically genetic engineering, and they are designed with essential features like the IGK leader, the histidine tag for protein purification and also for protein identification. Then the binding domain, which is the H22 SCFV, was designed, and also the SNAP tag, which is used for binding of the BG modified substrate. Then the SNAP tag, being a human protein, is being expressed in mammalian HEC293T. The recovered protein from the mammalian cultures were purified using an affinity purifier. They immobilized metal-ion affinity and size exclusion. Then after that, the protein, the full-length protein, have been structurally validated using a combination of SDS page and Western blots, after which the protein are being conjugated with the BG modified substrate to generate either the ADC or the PIC. Also to evaluate the SNAP conjugates on cell sample, monocytes are being recovered from donor blood, and they are being allowed to differentiate into macrophage over a period of 7 days, after which they are being polarized using cytokines into M1 and M2. And these are being compared with the M0 as the resting. There's functional assay, including flow cytometry, internalization, and cytotoxicity were carried out. So I will just be sharing an overview of some of my scientific results. After retrieval and protein purification, the full-length protein of H22 SNAP was confirmed using SDS page and Western blot, as I said earlier, at 54 kilodaltons, and using the BG modified fluorophore BG Alexa 488, we are able to confirm the functionality of the SNAP tag domain. After that, the SNAP fusion protein the binding study was carried out on a CD64 positive U937 monocytic cell to show receptor binding, and we observe a positive receptor binding up to 80%, more than 80% receptor binding, which shows that this can effectively be used for binding and also cytotoxicity. Also, the macrophage that was retrieved from donor samples were characterized phenotypically. And first, I used confocal microscopy to show a difference in the morphology, and the polarized M1 macrophages was showing an elongated shape. However, the M0 and M2 were showing an extensive shape, which was also confirmed in literature. Next, we showed using flow cytometry that there is a CD64 receptor overexpression, especially with macrophages that have been stimulated with interferon gamma and LPS. And this was observed in the M1 stimulated macrophages, and also in the M2 stimulated macrophages with 98% CD64 and also 79.4 CD64 in M1 and M2 macrophages. However, in the non-stimulated macrophages, which is the MO, we have a baseline expression of CD64 at around 49%. Next, we used confocal microscopy to show the internalization kinetics of the CD64 specific SNAP tag immunoconjugates. And as I've shown previously in the CD64 flow cytometry, this also confirms that all the macrophages internalize the CD64 specific SNAP tag immunoconjugate. However, the internalization is dependent on the CD64, as the M1 macrophages shows the highest internalization, followed by M2 and M0. Lastly, in the results section, I was able to show that the ADC generated, which contains the H22 SNAP conjugated to the BG auristatin F, demonstrates selective in vitro cytotoxicity in CD64 positive monocytic cell lines, but it wasn't demonstrating cytotoxicity on a CD64 negative cell line, the A2058. And this actually shows that the antibody drug conjugate can selectively kill a CD64 positive monocytic cell in vitro. Also, although the result is not shown here, the ADC wasn't used in this case for ex vivo macrophages because our ADC is actually used for killing proliferating cells as an antimitotic agent. So we used the photoimmunoconjugate, which contains the H92SCFV SNAP engineered with the IR700, and under visible light, this exact CD64 dependent phototoxicity on ex vivo polarized macrophages, as we can observe from this at all concentration tested 20 nanomolar, 100 nanomolar, 500 nanomolar. The highest killing was observed in M1 and comparable to M0 and M2. And why is this very important? This is important because depending on the deceased model, the PIC can be a good clinical agent. For example, for M1, that can be used for treatment of chronic inflammatory diseases, and for M2, for the treatment of cancer. And for M0, the PIC can also be used for the treatment of certain infectious diseases, whether in the activated form or in the non-activated form for macrophage mediated diseases. So from this research, we are able to show that cytokine-stimulated macrophages show overexpress CD64 receptors compared to resting macrophages. Also, the SNAPTAG technology presents a targeted immunodiagnostic or therapeutic approach for disease treatment. Also, this targeted elimination of dysfunctional cells depend on CD64 overexpression, which is specific to the diseased models. And finally, that the CD64 is a pivotal therapeutic target, and anti-CD64 SNAPTAG immunoconjugate could be potential precision diagnostic and therapeutic agents against monocytes or macrophage-driven chronic diseases. As a means of a treatment implication, the ADC could be delivered localized or in systemic delivery in hematological cancers like the AML, and in certain skin cancer like melanoma. And also for the photoimmunoconjugates that can be delivered locally or systemically in solid tumor or chronic skin lesion like chronic inflammatory skin disease or atopic dermatitis. Acknowledgements to my PI, Professor Stephen Bart, and my co-supervisor, Dr. Alex, and every member of my research unit at UCT, and also the funding body, including the NRF and the MasterCard. Thank you, everyone, for listening to my presentation.
Thank you indeed, Emmanuel, for the great presentation. One question from our side. What do you see as the advantages of the SNAPTAG antibody and technology approach compared with other targeting strategies?
Okay, thank you very much. I think for the SNAPTAG technology, this technology, it's a very unique technology that actually allows for the binding and also the internalization of both a diagnostic and a therapeutic agent. For example, I cited the example of the R700, which actually the R700 as it is can be used for diagnostic and also for treatment as a single agent. And that is not possible with other targeted therapies. For example, as the part of previous targeted therapies is the recombinant immunotoxin. So this immunotoxin actually contains an antibody linked to a toxin. And in itself, the fact that it's a toxin actually has its own limitation, especially in terms of clinical, efficacy, clinical side effect, especially with patients' use.